In this video, we will discuss about the advantage of this Neoadjuvant PD-L1 blockade that will result in enhancing this cancer immunotherapy even before injecting any additional drug to enhance this immunotherapy. So how this Neoadjuvant PD-1 blockade works, its mechanism is shown in this figure. So here's the primary tumor site. It is shown here in the lungs, and the zoom in view shows that it contains the tumor cells, and it also has some this dendritic cells and the T cells. So when this anti-PD-L1 and uh, in the tumor microenvironment, these dendritic cells and T cells that are expressed usually have this PD-L1 ligand, so it can easily be deactivated by the tumor cells through uh, PD-L1 receptors that are expressed on the tumor cells. So if those interactions can be reduced through this anti-PD-L1, so this anti-PD-L1 will bind to the T cells PD-L1 ligand and the dendritic cells PD-L1 ligand and prevent the interactions of the tumor cells PD-L1 receptors to this PD-L1 ligand that are expressed on the T cells and dendritic cells, and thereby deactivating of these dendritic cells and the T cells will be reduced. And once this deactivation is reduced, then the dendritic cells and T cells can proliferate even within the tumor microenvironment. And during this process, what will happen is that dendritic cells will be able to provide informations, these tumor antigen informations to the T cells more efficiently, as the number of dendritic cells and T cells are high in this case compared to the case where there is no anti-PD-L1 ligand. And since more activations of the T cells occurs through these dendritic cells by providing informations of the tumor antigen, is more killing of these tumor cells through the T cells can occur. So this is one way that this neoadjuvant PD-L1 blockade will increase the tumor efficacies or uh, anti-tumor efficacies or this tumor killing by the T cells. However, there is additional effect that can also occur. So what this figure suggests is that some of these dendritic cells will be able to move into this tumor draining lymph node through this afferent lymphatic, and since the dendritic cells already has the information about the tumors, then it can activate the naive T cells that are present in this draining lymph node, tumor draining lymph node, and thereby activating additional T cells outside the primary tumor sites. And these activated T cells will then be able to reach to the primary tumor sites through this uh, afferent lymphatic and bloodstream, and this will further enhance the anti-tumor efficacies or the tumor cell killings in the primary tumor site. So this mechanism occurs even before any additional drugs is used for the immunotherapies such as CAR T cells or any chemotherapies that is done to kill the tumors. So this is the advantage of this neoadjuvant PD-L1 blockade even before the any kind of immunotherapies to enhance the anti-tumor efficacies using the host immune system. So one of the requirement is that there should be some tumor infiltrating lymphocytes present within the patient or in the tumor microenvironment, as then only it will be able to anti-PD-L1 will be able to enhance the anti-tumor efficacies. So this is we can say that dual mechanisms or dual mechanisms by which this neoadjuvant anti-PD-L1 blockade enhance the immunotherapies. First one is by enhancing the activated T cells within the tumor microenvironment, and second one is through the activations of the T cells outside the tumor microenvironment. And here the micro metastate in tissues is shown here. So not only the activated T cells will be able to reach to the tumor microenvironment or the primary tumor sites, but it might also goes into this micrometastate in tissues, thereby keeping the T cells ready for any future, keeping the T cells with the informations about the tumor antigens preserved for the future tumor cells tumor cells regrowth, or the tumor cells regrowth at for the tumor cells regrowth at other site because of the proliferations or migrations. This is how this neoadjuvant PD-L1 blockade helps in cancer immunotherapies. So that's all for this video. If you need to understand more about these mechanisms in detail, you can go through the reference that has been provided in the description. Thank you for your time.